Hi, I'm Mr. Baker. This is Unit 2, the Chemistry of Life, or Biochemistry, Section 2.5, which covers proteins, enzymes, nucleic acids, which is DNA and RNA. But first, proteins. They are the chief building blocks of life. The alphabet that makes up protein are called the amino acids. Those are the monomers of proteins. The protein itself is the long chain of amino acids, or the polymer. There are 20 unique amino acids, but you can put these together in all kinds of different combinations for unique chains. There are 20 different amino acids, and each has a different R group that makes them unique. So for example here, they have the side chain, the amino group at the top, the carboxyl group at the bottom, and the side chain. It's the side chain that's different. Those are called R groups, and there are 20 of them. Your body can produce 11 of the 20 amino acids on its own. The other 9 essential amino acids must come from your diet. And you will eat them in various foods, and there is a list of them. There are 4 levels to the protein structure. 3 of them you will have, and the 4th you might have, depending. Primary, gotta have it. Secondary, gotta have it. Tertiary, gotta have it. Quaternary, you might have it. Primary structure is the sequence of amino acids. These amino acids are joined together via peptide bonds. In the bottom of the picture there, you've got, I'm going to guess, 35 amino acids joined together. That takes us to the secondary. Secondary has other what are called motifs, but the 2 big ones you need to know about is an alpha helix. That little squiggle at the front of helix is an alpha, and that is a spiral, like half of a spiral staircase. The next one is the beta sheet, and that weird looking B is beta, and the alpha and beta are Greek, of course. The beta sheet, those look like an accordion folded piece of paper. So you have a helix and an accordion folded piece of paper. The tertiary structure is how those helices and sheets form together with even other motifs, and we're going to call that a blob of a protein. And you could be done with tertiary structure, but some, you need more than one blob for it to work. So that is quaternary. Quat coming from quad, meaning 4th. It's the 4th level. This is hemoglobin, pictured at the bottom right, and it is 4 blobs put together. So hemoglobin does have quaternary structure, but not all proteins do. So here's a nice little GIF. Primary structure, their secondary structure with the helix and the sheet. The tertiary structure gives you a blob, and then a blob, more than one blob is the quaternary structure. The shape's going to tell it what it does. That's the shape of the protein determines its function. The proteins don't die, they don't get killed, they get denatured. Denature means that the protein has fallen apart. So it means if you needed it to be in 4 different blobs for quaternary structure and they come apart, it's been denatured. If you need it to be a certain blob and the blob falls apart into alpha helices and beta sheets, that's denatured. It can be caused by strong acids, strong bases, concentrated inorganic organic solvents. The big one that you should know about is heat, because if you boil your water, you've made it sterile, meaning you can drink it, and that will protect you against pathogens. So, by adding in heat, you can kill things, meaning you denature their proteins. Some of the most important things about proteins is that they can be enzymes, and it's a molecule that helps initiate and accelerate chemical reactions. They are not used up, and they remain exactly the same. You will need to know these terms and how they apply to enzymes, which is substrate, product, and active site. The substrate are the reactants. The actant site is the location on the enzyme where the substrate binds. There are 2 ways how they work. The lock and key, which is the top GIF, and then the induced fit, which is the bottom one. And you can see how they sort of work. Induced means it comes in and squeezes it. Lock and key is like an actual lock and key. Your product is released. Here are some more enzymes doing their work. Just let you watch these quickly. And then here are some other ones with the induced fit. in the lock and key. Here are some more enzymes doing their work. Looks like the one at the top, well, it's changing, so. And then here are some static pictures which aren't as good. Lactose intolerance is when people have a mutation to an enzyme that doesn't let them digest lactose, which is milk sugar. So those people have a difficult time eating dairy products. Nucleic acids carry the genetic information. Their monomers are called nucleotides. You get two types of nucleic acids, DNA and RNA. They carry the genetic information in large pieces of DNA that are called chromosomes, which are DNA and the proteins that hold them together. A gene is a small segment of DNA that codes for a particular protein or a particular piece of it. Yeah, we'll just go with protein. These nucleic acids control the cell processes. The DNA is the blueprints of the cell. The RNA carries out these instructions. So the RNA is the copy that goes out, where DNA is the master copy. A building block is the nucleotide. The nucleotide is three main parts: a phosphate, a sugar, there's your sugar, at the bottom, your phosphate, and then a nitrogenous base. Nucleic acids, A will bind to T, C will bind to G. You'll need to know the four bases: adenine, thymine, guanine, and cytosine. The base means that it just has a pH of over 7. DNA will stay in the nucleus. It's referred to as deoxyribose nucleic acid. The shape was elucidated, which is a fancy word for meaning figured out, by Watson and Crick in 1953, and it is a spiral staircase with steps, or what is called a double helix. So from the cell to the nucleus to the chromosome, down all the way down to the DNA. So here's another picture of the DNA. It's a double helix, spiral staircase. The steps are the two bases. RNA is a single helix. It's half of a spiral staircase with half of a step. It has A and U, no T. Thymine is gone. It uses uracil instead, and it does have G's and C's. It's made in the nucleus, and it travels to the cytoplasm or the ribosome for protein synthesis. Its sugar is ribose instead of deoxyribose. Here is DNA versus RNA and their differences. Don't forget, you have U instead of T. You have a single helix instead of a double helix. This is a table I've made that should be available on your topic page, and that will finish it. That's quiz 2.5 covering proteins, enzyme, nucleic acids, DNA, and RNA. Study hard.